Welcome everybody. Hello and thank you for joining us today for the webinar hosted by ISEH. My name is Simon Haas and I'm a junior group leader at HiStem focusing on single-cell systems analysis of stem cells at the interface of hematopoiesis, immunology and cancer. I'll be your moderator for today's session, which is entitled Tracing Single-Cell Fates in Hematopoiesis, which is actually a follow-up from the 2019 New Investigator tech session. In this webinar, we will hear from 3 technical experts about the most powerful methods available to trace cell fates in the hematopoietic system. This webinar will cover the basic methodologies, but also in particular the technical considerations for single-cell transplantation assays, lentiviral barcoding technologies, and sleeping beauty barcoding. If you have any questions at a time during the presentation, please type your question into the question box or into the chat box. We will have time for questions at the end of the webinar, but you're welcome to enter your questions into the chat box at any time during the presentation. It is now my privilege to introduce our first speakers of today's session, Dr. Dawn Lin. Dawn Lin recently completed her PhD and is currently a postdoctoral researcher in the laboratory of Sherlyn Naik at WEHI in Melbourne. Her research focuses on using single-cell lineage tracing and single-cell RNA sequencing to understand the heterogeneity of hematopoiesis, in particular the development of dendritic cells. Thank you for staying awake so long. It's now 1 a.m. in Australia, so thanks for staying awake, and we're really looking forward to your talk.
Thank you very much for the introduction, and today I'm going to talk about the applications of ex vivo lentiviral barcoding in my talk, and specifically give 2 examples of how I use this technology in my research. Oops. To start with, I'm going to go through the basic workflow of cellular barcoding in the lab. So in our lab, we have a lentiviral barcode library containing of approximately 2,500 different DNA barcodes. So each barcode is basically a short stretch of DNAs that are random nucleotides, and each of them are different to each other. So the first step is to label cells with barcodes via a common lentiviral transduction protocol. And there are 2 specific things that we should be careful about. The first is that we're aiming for a very low transduction efficiency so that to minimize multiple integration, that is multiple barcodes enter the same cell. And the second one is to use a low percentage of the total library into each biological samples so that to minimize a repeat use of barcodes. That is the same barcodes labeling 2 different cells. So after we barcode label the cells of interest, we can then either culture the cells in any culture condition of interest or transplant them in vivo to allow development. And after a period of time, we can then isolate the progenitor cells populations, usually via FACS sorting. And next, we can then perform PCR to amplify the barcodes and sequence them. And the analysis will be to compare the presence or absence of barcodes in a different progeny populations. For example, if you look at the blue barcode here, which is present in all 3 different populations, this implies the original barcoded progenitor cells have a multi-lineage output. Whereas if you look at the different barcode, which is only found in the second population but not present in the other populations, this implied a restricted output from that progenitors. So the first question that I'm interested in asking is to use barcoding to study clonal dynamics. So from many recent single-cell tracking studies, we now know that phenotypically defined multipotent progenitor cells are actually very heterogeneous in terms of their fate output. That is, single cells can have a very strong bias towards one or the other cell types, and only a handful of them are truly multipotent. But a lot of those studies have a major limitation in that they only measure clonal output at a single time point. But if you imagine a scenario in that a clone can give rise to different cell types at different time points, and if you measure this clonal output at different time points, you can get a different conclusion. So we think that it's important to measure clonal dynamics over time. So to do that, I use a workflow that is very similar to what I just described. Basically, we barcode label the stem progenitor cells for the bone marrow. We then put the barcode label cells into a culture system that allowed the generation of different dendritic cell subsets, which are the cell types that we're interested in. We then perform serial sampling to allow the measurement of clonal dynamics over time. So serial sampling is basically at each time point we mix the cells in the wells and remove half of the content for antibody staining and FACS sorting of different subsets. And the other half was kept in culture with freshly added media for further development. We then repeat this process every 2 days. And after that, we've sorted all the cell types over all the different time points. We can then amplify the barcodes and analyze them. So it is important to perform split controls at each time point to make sure that the time interval, at each of the time interval, when you remove 50% of the content, it is representative of the other 50%. So we perform that for all the different time points that we do in the real experiment and compare the similarities between those split controls. As you can see, they're highly similar, which means that our approach is pretty fair in terms of measuring clonal dynamics. So the next thing we wanted to do is to visualize clonal dynamics over time. As you can imagine, there are a few major challenges in doing so. First of all, in a single experiment, we can track the development of thousands of different clones, and each of them have different phase distributions, meaning that they can generate different subtypes in different proportions. They might also differ significantly in terms of how many cells they can produce. And most importantly, all these properties can change over time within different clones. So to overcome these challenges, we have, together with our collaborator Jerry Gao and Andre Kant, we have developed a technique called developmental interpolated Disney time-lapse movies, in short Disney movie. So the first step is to use, to apply a Disney, which is a dimensionality reduction technique, to allow us to look at all these different clones in a 2D plot. So Disney will place clones with similar properties close together to form clusters, and next to visualize the different phase distributions, instead of each dot representing a clone, we plot each dot as a pie chart, and the proportion of the different slices represents the proportional output to different cell populations. And we also scale the size of the pie according to the size of the clone to visualize clonal size. And next, we then link the different time points together to visualize the clonal dynamics over time. As you can see, different clones changes in their size as well as their contribution to different cell types over time. And we can also see holistically a rise and fall of different clones that contribute to different DC generation. So the next step is to understand the underlying patterns of the data set. So to do that, we apply clustering on the Disney, which identify different clusters. And here I'm showing two different, very distinct clusters. So cluster 7 shown here generate large numbers of CDC1 and CDC2, which are the conventional DC subsets, whereas cluster 12 here did not make a lot of CDCs but generate a different subtype of dendritic cells called the plasmacytoid DCs. And if we look at all the different clusters and comparing their patterns, we can then group the different clusters into 3 major classes. The first one, which is highlighted in red here, are the real multipotent clones that can generate all 3 disease subsets. There are also a large number of CDC biased clones, meaning that these ones make a lot of CDCs but very little PDCs, whereas the bottom ones make predominantly PDCs but very little CDCs. So to summarize this part of the talk, we have developed a framework using cellular barcoding to allow us to characterize the different clonal dynamics, which include the following properties. That is, what type of cells a clone to make and how many cells a clone can make and when to make them. And using this framework, we have, to understand DC development, we have identified multiple major classes of DC trajectories, from hematopoietic stem progenitor cells including the multipotent ones as well as the CDC or PDC bias clones. So the next question that we use cellular barcoding for is to ask the clonal etiology of perturbed hematopoiesis. The system that we are interested in is to use a cytokine called FIC3 ligand, which preferentially increase dendritic cell development in vivo. So what we did was to barcode label stem and progenitor cells, transplant them in vivo into recipient mice, and they will then receive either FIC3 ligand to increase DC generation or PBS as control. We then harvest the spleen and different progeny populations, sorted them and analyzed the barcodes in different populations. And as you can see, we see a very preferential increase in CDC1 numbers, but very pretty much unchanged number in other myeloid and B cell populations. And the question we really want to ask is that what accounts for that increased expansion of DCs? So if you imagine that in a steady state we have 4 DC being generated from the hematopoietic stem and progenitor cells, and the first possibility can be that the cytokine purely just enhance the expansion of pre-existing clones, and if that is the case, we will expect to see increased number of DCs but similar number of barcodes that contribute to the DC generation. On the other hand, if there is a second possibility, can be that the cytokine induce recruitment of dormant progenitors that would otherwise do nothing. And if that is the case, we will expect to see an increased number of barcodes found in these DC populations. And there is also a third possibility, which if the cytokine can induce a divergence of different clones, meaning that these clones were meant to make a different cell type in steady state, but upon the cytokine stimulation, they are now switched their fate into making DCs instead. If that is the case, we will expect to see not only an increased number of barcodes in the DC population, but also a decreased number of barcodes in a different population. So because for the interest of time, I'm not going to show any results, but so far is to say that our conclusion is mainly the cytokine FIC3 ligand mainly drive emergency DC generation via enhanced clonal expansion of pre-existing stem and progenitor cells. So the last slide, I just want to really mention this review that was published in Experimental Hematology in 2014, which outlines a lot of important considerations and caveats or limitations that you should think about when you applied ex vivo lentiviral barcoding in your experiment. So I really encourage you to check it out. So with that, I'm going to thank all the people that helped to do my work during my PhD, particularly my supervisor Charlene Naik, and also my collaborator Andre Kahn and Jerry Gao. Thank you very much.
Thank you for this great talk. I really enjoyed it. So I'm now pleased to introduce our second speaker for today, Joana Carrelha. Joana is a postdoctoral researcher and in the lab of Sten Erik Jacobsen in the Heidelberg Institute of Molecular Medicine at the University of Oxford. She obtained her bachelor and master degree in molecular biology and genetics from the University of Lisbon and obtained her PhD in clinical laboratory sciences from the University of Oxford. And in the recent years, she has been focusing on the study of clonal properties and differentiation mechanisms of marine hemolytic stem and progenitor cells, with a particular focus on megakaryocyte lineage differentiation. And she will now talk about single-cell transplantation assays to study hematopoiesis, and I'm very much looking forward to her talk. Thanks.
Thank you very much for the introduction, and thanks to everyone for joining to hear us talk today about these techniques. I'm Joana, and I will talk about the technique of single-cell transplantation to analyze the functional heterogeneity of stem cells, and in my work I focus on mouse hematopoietic stem cells. So this is a technique that we used recently to describe different functional types of stem cells from the bone marrow of mice based on their in vivo output and the kinetics of their output. So the technique is described there if you want to have a closer look at the methods. I think everybody is quite familiar with the general concept of transplantation of bone marrow or hematopoietic stem cells. And this technique has the same concept, the difference being that single stem cells from the bone marrow of a mouse are deposited directly into plates with media, and we add a limited number of whole bone marrow support cells, and then we transplant that directly into conditioned recipients. I use lethally irradiated mice, but you can conceive of different types of conditioning based on your experimental purposes. Also, I will describe a bit the different types of modifications that you could make to this protocol for your purposes if you would like to implement this technique, and especially the technical considerations when you are thinking of modifying the technique. So I won't focus too much on data, but rather on the technical considerations. So first off, the first step will be the sorting. So I sort the single cells directly into U-bottom 96 well plates with 100 microliters of media, and then I usually use a very limited number of unfractionated support cells, around 200,000. And here it's very important to have a reliable way of counting the cells, because your recipient mortality can go up quite dramatically if you don't provide enough support cells to the mouse together with your single cell of interest. You need a sorter with automatic cell deposition, so that could fit different types of plates. And then for your purposes, depending on which cells you want to sort, but for hematopoietic stem cells from the bone marrow of mice, I find that the 85 micron nozzle is quite good. There's a good compromise between high speed, but with a pressure that is still acceptable for good viability of the cells. Something that you might consider optimizing for your particular machine is the phase mask. So different sorters come with default single cell modes, but that mode might actually be a bit too strict. So if you want to reduce the number of target cells that are discarded by the machine, you might want to test and find a less strict phase mask that will allow you to still accurately sort single cells and deposit single cells, but with a much higher yield that will save you some time and some cells. You want to minimize the processing steps, the actual steps, and also the time that you take with each step. So I wouldn't overcomplicate the protocol unless it really is needed experimentally. Usually enrichment before the single cell sort is not really necessary, but if it is necessary for your purposes, you might consider an enrichment with microbeads or a depletion or even a yield sort. But I would only do this if it's really required for the feasibility of your experiment. Otherwise, I would minimize any pre-single cell sort processing steps. I usually just sort the cells into plates and keep them on ice while loading, but you could, if you are interested, modify it in a way that you incubate the sorted cells with some experimental conditions before you inject them. That's possible, but you always need to consider the effects of any protocol variation. So you might be reducing the engraftment frequency and the quality, the type of engraftment that you get, depending on the conditions that you are using. Also, another consideration is the phenotypic definition that you use for your hematopoietic stem cells. Of course, there's different possibilities, and you might want to modify it for your own purposes. But we have found that when you change your gating strategy, you can find that you improve certain parameters while modifying others. So that's something you need to keep in mind. So you might improve the frequency of long-term reconstituted mice, but you will change the frequency of the different types of reconstitution that you might get, as I tried to exemplify here. If you sort SCA high cells, you will improve the number of reconstituted mice that you get, but you will preferentially select for platelet biased patterns, and you will preferentially lose robust multi-lineage patterns. So that's something to think about. You can always index your sorts, your cells, so that you can acquire a bit more information about the cell surface markers. And it doesn't need to be a marker that you're gating on, but as long as it's in the staining, you will be able to index for it and then retrospectively analyze it. And I just like to mention that this technique works with stem cells from the bone marrow, but also with fetal liver and fetal bone marrow cells. Although at the moment we obtain quite a bit lower engraftment frequencies compared to the adult bone marrow. Another thing to consider is the recipients. So this is a technique that requires quite several independent cohorts of recipients, so separate experiments from several donors because of the variability of mice that you get. You can statistically try to calculate how many types of reconstitution you will get, but there will be biological variation between experimental days. And you will require quite a lot of mice for most purposes, but this will depend on your hypothesis and the type of analysis and the engraftment frequency that you obtain. My typical cohort size is 40 to 60 recipients. One thing that is quite nice is that you can use many different mouse phenotypes, either inducible or constitutive, as your donors or your recipients. And I'll talk about this a bit more, but you can use fluorescent reporters, mice that harbor mutations, they can be immune compromised. So you can test a lot of different things, but beware that when you have mixed genetic backgrounds, you can have immunological compatibility issues. So you might have early engraftment loss in certain cases. So these combinations are something that needs to be tested. The types of downstream readouts we can do, there are several at the functional level and molecular level. So each recipient encapsulates the activity of a single stem cell that was transplanted, and the properties of each subtype can then be further tested by testing the donor-derived cells. So I mainly focus on flow cytometry, but there's other types of assays that you can do. Look at the reconstitution in the mice of the progenitors and mature cells. You can have something that's very nice in this technique is that you can do serial testing, so you don't need to destroy your recipient in order to have a lot of readouts. So you can have a kinetics over time. And then you can sort any donor-derived cell type for molecular techniques, for in vitro cultures, for in vivo assays, for imaging, and other types of assays. In terms of the reconstitution readouts, I would always recommend to base your analysis on positive markers rather than negative. So if you look for a GFP positive cell in a GFP negative recipient, that's typically much more sensitive and accurate than doing the opposite. So the background in your fluorescent reporter recipients is something that is very important in your setup. So you have to look at all your different populations of interest and see if there is any cells that you were expecting that were labeled and they are actually not, because you need to use that as to set your thresholds for positive reconstitution. And this really needs to be done per individual recipient type. I would really recommend trying to find the best conjugate combinations for your panels for both sorting and the readouts. The good separation between donor-derived and non-donor-derived cells is really essential. And as I mentioned, you need appropriate and reasonable detection thresholds. So take that into consideration when selecting your donor type and your recipient type. In terms of flow cytometry especially, I would like to point out that we find that blood platelets often stick to other cells during sample processing. So if you're using fluorescent reporters that also label platelets, it's important that for each population you are able to distinguish between real donor-derived fluorescent signal and fluorescent signal that actually is coming from labeled platelets. So your cells of interest might not be labeled, but if there is a platelet attached to them that is labeled, you might have a positive, a false positive. So you need to take that into consideration, especially in blood readouts. So this technique is a very powerful method to isolate the HSC subtypes for further testing. So as I mentioned, in each mouse, all donor-derived cells come from the single HSC subtype, and then you can combine the technique with other fate mapping techniques. And I have a few examples here. So you could combine it with constitutive or inducible Cree fate mapping, and you can induce the Cree labeling before or after transplantation, depending on your purposes. So you just need your donor to be the appropriate mouse model that you need. You could also combine it with cell cycle analysis and proliferation tracking methods. so live cell staining or genetic mouse models. And again, you can have this in before the single cell transplantation or after, depending on your purposes. And another option is also you can combine it even with another technique that is being talked about today, which is the DNA barcoding. So you can start off with single cell transplantation, then you sort out your donor-derived cells and barcode them for a more high approach in represented in the reconstituted mouse. With careful experimental setup and appropriate controls, the output of your stem cell is mostly unambiguous, so you can quite carefully and quite accurately describe the output of that stem cell. Something that I think is a big advantage is that the donor-derived output can be visualized and quantified by serial sampling of the mice throughout time. So before using any destructive methods, you can very flexibly have your experimental decisions based on your ongoing observations of the results. And also, as I mentioned, this technique can be combined with many different types of readout methods and also other fate mapping techniques for more complex type of analysis. But of course, it has disadvantages and its limitations. It is a transplantation setting, so we're not looking at native or unperturbed hematopoiesis. Any non-engrafting stem cells you will not be able to analyze at all, so you are losing that information. It's virtually impossible to discern if you don't have engraftment because of technical factors in processing or because your cell didn't really engraft. And also if the engraftment level is very low, you might not be able to detect that stem cell actually engrafted. But depending on your experimental setup, the phenotypes of your donor and recipient, and your detection thresholds. And of course, robust data requires using quite large numbers of mice, and maintaining and analyzing these large cohorts can be quite time-consuming. Also, as I mentioned, this technique is not always directly applicable with specific donor-recipient combinations. So whenever you're modifying the protocol, you will need substantial testing to make sure that everything works properly. So that's my summary of the technique, and I would just like to acknowledge my PI, Sten Erik, and both our labs in Oxford and in Stockholm, and all of our collaborators here in Oxford and in Stockholm.
All right, thank you very much, Joana, for this really great and broad overview about single-cell transplantation technologies. I have now the pleasure to introduce our third and final speaker today, Dr. Alejo Rodriguez-Fraticelli. Alejo completed his PhD at the Autonomous University of Madrid, working on cell polarity and epithelial morphogenesis. He then joined the laboratory of Fernando Camargo at Harvard, where he has developed genetic lineage tracing methods to study hematopoiesis, specializing in single-cell technologies and novel approaches to cellular barcoding. His early work as a fellow uncovered the functional heterogeneity of hemolytic stem cells in situ, but currently is focusing on the application of genetic lineage tracing methodologies to really understand the pre-malignant stem cell dysfunction and improve diagnosis and prevention of blood disorders. So we're really looking forward to an exciting talk. You were welcome.
Thank you so much, Simon. And basically, this is the introduction. Thank you so much for this introduction. What I'm going to talk to you about is this method for inducible transposition to track native hematopoiesis. And essentially, it's a method to address this question of how do progenitors decide what they will become. And it's, in general, all these lineage tracing and single cell tracing methods are trying to get to that question of heterogeneity and how cells end up deciding what type of differentiated cell in the blood they're going to make. And basically, you know, after 140 years of molecular biology since Pappenheim's first description of the stem cell lineage tree in the blood, we basically have this new vision that this process is actually way more complex and way more plastic, and there's extensive heterogeneity already at the stem cell and progenitor levels, where cells that were supposed to be multipotent or bipotent are actually highly diverse in their capacity to generate one or each specific lineage. And so the actual clonal tracing is actually at the origin of the study of stem cells. And already from the original description from this stem cell property in the blood system by Thiele and McCullough, they used a method for tracing HSCs in order to describe that actually all of these different types of progeny in these spleen colonies were actually being formed by a single cell. And obviously we have much more advanced methods now, but I just want to sort of bring it back that this is a tool or a set of tools that is really at the origin of the study of these incredible populations. Now, what we try to do with in the lab is actually apply methods for multiplex clonal tracing that allows us to track multiple clones inside one single mouse. That's very different from this single cell transplantation and more akin to these DNA barcoding methods that Don was talking about earlier. The advantages are obvious. You can trace many more clones inside a single mouse. You also require fewer animals, and there's obviously, because of that, less clone-to-clone variation because these clones are being tracked inside the same subject. Now, obviously, the challenges for these multiplex methods are that they are much more complicated to set up in your lab. They're also a lot more expensive, typically involving some version of high-throughput sequencing. It also has, and this is something that is barely talked about, but they are actually plagued by false negatives and false positives, some of them including that review that Don was suggesting to read. This is a very important consideration as a technique. You will typically have a lot more false negatives in these multiplex tracing methods. Another challenge is that there's obviously less literature. They're much newer. So there's fewer studies and less methodological consensus, although there's increasingly a lot of effort towards this. And finally, they are a little bit subject to this technology bias, the fact that there is a lot of innovation in this field and thus very little depth sometimes into some of these technologies, which are replaced every year almost. So, but this is something that we expect will be solved with time. Now, the type of experiments that we do for Sleeping Beauty transposon. So the way that the Sleeping Beauty transposon barcoding mouse works is as follows. We start with a RTTA that is constitutively expressed that allows us to control gene expression using doxycycline. And we then have a knocking allele in the collagen locus that expresses the Sleeping Beauty transposase in response to this doxycycline. So we feed mice doxycycline to activate the expression of the Sleeping Beauty transposase. And then we have a second knocking in the collagen locus, which is the cognate transposon, which will be excised by expression of this transposase from this, what we call the donor locus, and integrated into a semi-random place in the genome of each cell. And now, if we try to recap the clone tracing methods that we just talked about, so receptor polymorphisms or fluorescent proteins as reporters for single cell transplant, and lentiviral DNA barcodes, the endogenous transposon integration system has two main advantages compared to these methods. The first one and most important one is because it's an endogenous system in the mouse, it's encoded in this loci that I just explained. You can actually trace clones without isolating the cells from their native niche, and therefore the perturbation level is much lower. And it also has the added advantage that because the integration is semi-random in the entire genome, the diversity, the maximum diversity of the system actually allows you to track millions of clones at once, and essentially in a completely random process. So these two advantages are what allows us to design the following type of experiments where we can trace the origin of cells without isolating prospective progenitor stem cell populations and then actually look at their clonal relationships. So for these experiments, we typically start with a mouse in which all the cells have the transposon in the donor locus. After doxycycline addition, the cells are essentially uniquely barcoded with a transposon integrated in a different site in each cell. And then we do this type of post-chase experiment in which we remove dogs and then we let the barcodes from progenitors slowly start replacing the downstream progeny, and therefore progeny that have a similar clonal origin or shared clonal origins will begin inheriting the same barcodes. Okay, So you'll start seeing shared barcodes between populations. And then we isolate this progeny of interest, typically by fax, and we can then proceed to prepare libraries for cloning and sequencing these transposon integration sites, which are typically analyzed by high throughput sequencing. So the method has, a lot of advantages. I obviously talked a little bit about the fact that it's an in situ method, so it allows you to look at native unperturbed hematopoiesis, and that remains actually one of the major advantages of the model. I also talked a little bit about this fact that it's, and finally, it's also a relatively unperturbed system, not only because of the fact that you are tracing cells in situ, but also because it's a single copy transposon. So even though the integration might affect a gene, since it's a single copy, it's very rare that you will get a significant perturbation in the cells that will lead to the appearance of some malformations or some defects. Typically, these integrations might lead to the death of the clone if it affects a cell that is required for viability of the cell, and that is just lost from the system. But it's a very rare process as far as we observed. Now, the disadvantages of the method are also very important to note. We still, at the current system, with the best available transposases and transposons, we actually don't see transposition in about 60% of the cells, even after months of labeling. The method depends on sorting purity and obviously availability of gDNA, so you can do it from erythrocytes or platelets, which don't have a genomic DNA, but also only nucleated cells. The fact that you need to analyze it with high throughput sequencing means that you end up spending a lot of money and resources on sequencing these samples. And finally, because of this large diversity and also because of the techniques for sequencing these transposon integration sites, it's a method that typically tends to undersample, and it's plagued by false negatives. So what can we do with this type of powerful but yet very sparse data? This is sort of the flow of information that you would want to obtain from this experiment. You would start from the left with your raw data, typically analyzed as a table or a heat map, as here, with the clones in the rows and the populations in the columns. Then you would sort of try to guess what's the distance between any two of these populations to try to find some common origins or some structure in how these progenitors are generating these cells. And then you would end at the other end of the analysis with your interpretation of what are the relationships between these different cell types. Now, I'm just going to take you briefly through what we do in order to get there. So basically, we start typically with the raw data here. You obviously can't find much information. This is just the raw data as is obtained from the sequencer, pretty much. You can scale adjust it to try to label those clones that are a little bit smaller in size. You can also try to binarize the data, so true, false, do we detect the clone or not in that population, to try to get something with a clearer view. But typically what we end up doing with this data is try to normalize it by population. So just plotting the fraction of each clone within each population, 0 to 100%. And from this normalized raw data, we typically have to filter contaminating clones. This is clones that can appear in multiple mice because of recurrent integration sites, which is something that does happen in these mice. Also sometimes cross-sample contaminations. That's why we always try to sequence these experiments, always having two mice in the same sequencing run to try to remove these sort of cross-sample contaminations and also estimate sequencing errors. And finally, we do some auto-thresholding of this data to remove the clones with lowest reads. And this typically comes from analysis of split samples. So splitting their DNA into two and sequencing them at the same time. And also histogram-based thresholding can be used. Finally, to compare these populations, we need to use some sort of distance matrix between these two, between any two populations. And so these distance metrics, typically you can use correlations or other sorts of mathematical spatial distances. And then this can be plotted using dendrograms, which are a way to establish which two populations are closer in this particular space or metric space. And then another way of representing this that is also very useful is using a square form of this distance matrix. And in this particular case, I'm representing the 1 minus the distance using the correlation, and you can see that in this particular case, monocytes and B cells are have a much shorter distance and are more related to each other in this particular metric. Now, very importantly, different metrics actually provide very different information. Okay, so like correlation metrics, the ones that I was just showing you, are actually very strongly influenced by clone sizes. So if two populations share even a single clone that is the largest clone, they will have a very high correlation, despite actually not sharing the majority of the clones in the data set. So on the counterpart, Boolean metrics that are just true-false metrics are actually very strongly disturbed by false positives, which can can be highly present in this type of high throughput sequencing analysis. So there's two metrics that we usually prefer in the lab to use. One is to use the ranked correlation or the Spearman correlation, and the other one is to calculate actually the observed versus expected ratio of shared barcodes. Usually calculating this from a null model where each progenitor is assumed to be independently arising from a stem cell. So, for instance, erythrocytes are only replaced by erythroid progenitors, basophils by basophil progenitors, and so on. And then calculating what's the observed overlap between any two populations over that expected null hypothesis. And obviously, you can use both correlation and this observed to expected ratio as a distance metric to plot these dendrograms. But obviously, these are not the real lineage tree maps, but just inferences of the process. So there's a couple of more things that I want to talk about and discuss related to these techniques. And the first question that I want to address is if we can actually use these type of techniques to know what a stem or progenitor cell generated without transplantation culture. It is actually quite difficult to do when you have populations where self-renewal is very low, such as, for instance, myeloid progenitor populations. The reason is that if a progenitor like this purple cell here does not self-renew, even though it's in this particular case bipotent, you will only detect the common origin because of this overlap between yellow and blue cells that I'm showing here, but you will rarely detect any overlap with the purple cells, or any of the purple barcodes. And this is because this population, since it's not self-renewing, those barcodes are exhausted at the time that these cells generate their progeny. So self-renewing is actually an essential property of the cells that we know exists in the bone marrow and actually allows us to track which type of progenitors they form. And in this particular case here, you can see that this purple cell is self-renewing over time and generating both blue and yellow cells, and you can detect those progenies because of this self-renewal and the fact that you can still see the barcode after weeks or months after analysis. So this is a very, very, very important conclusion from this type of techniques and very important technical assumption. Only self-renewing populations can be assayed without transplantation culture. Populations that have very low self-renewing, when you see any kind of overlap, it typically comes from higher self-renewing populations at the top of the tree. And finally, another thing that actually plagues this type of inference analysis is the fact that different populations have different kinetics. So in this particular case, I just drew a ground truth model here for this particular lineage map, where the red cell is generated at the very top of the tree, and then yellow and blue cells are generated at a second level, but those yellow cells are generated with a much lower flow rate. You can see that if you generate mock barcode data from this model, you will see a very high relationship between blue and red cells because they are the ones that are more quickly, more rapidly replaced. And then the interpretation of the model will actually always be to put them closer together and farther away from the yellow cell. So it's very important to understand that you need to accurately describe the flow rates of each pathway if you want to properly model the correct lineage tree. And these flow rates, if you know them, if you can calculate them using other methods or from the literature, they are very informative to establish a correct null equipotent model against which to test your observations. And obviously there's the problem that clonal heterogeneity might also affect the different flow rates, but that's a problem for the future actually. So can we actually find the true lineage tree? So one way that we can try to actually come with this, come to a little bit closer to a true lineage tree is actually to generate, or might actually be to generate, all the possible models for all the lineage trees that one can think of, modifying all the different parameters for these models, generate expected data, and then test those model data against our observations, and start with the models that you cannot reject. Now, this is obviously an exponentially complicated problem, computationally and from a modeling aspect, but I think that this will be the only way that we can actually find the true lineage tree. And more, we need more better description of the parameters to sort of narrow down the parameter space that we explore when generating these in silico models before we can actually start addressing this question at all. So with that, I would like to acknowledge the people in my lab that supported me, the funding agencies that support my work, and our collaborators, Caleb and Joana at the Klein Lab. And thank you so much for your time.
Alejo, thank you very much for this amazing talk. So we will now begin the question and answer portion of the presentation. So if you have any questions, please enter them into the question box or into the chat box. We already have the first questions, and the question we have is actually particularly addressing Joana. And the question is actually, what is the percentage of the recipients that can be successfully engrafted by using single-cell transplantation experiments? And the question comes from Peng Yi. So, Joana, do you have a good answer for this one?
Thanks for that question. So, it really depends on the type of donor and recipients that you are using, and also what parameters are you setting to analyze the reconstitution. So in my particular experiments, I use wild-type recipients, and I use double fluorescent reporter, and I look for long-term reconstitution, so that donor-derived cells must be above a certain threshold, which for me is 0.1%, at least 4 months after the transplantation. And in that case, I would say the average for most cohorts is around 40-45%. For specific gating strategies, you can go, I've gotten higher, up to 65%. But if you use immune compromised recipients, or a model like W41 recipients, you probably will get higher frequencies of reconstitution. And also, if you are also looking for transient reconstitution, you can get pretty close to 100%. Actually, the viability in these transplantations is pretty amazing. These cells, we've even sorted cells that flew, in the bone marrow flew over from Sweden in the morning and was injected in the afternoon, and there was the reconstitution percentages were as stable as ever. So it's actually quite amazing. But it really depends a little bit on your setup and your analysis parameters.
So conceptual question related to this. Do you actually believe that our current gating strategies are already, let's say, 100% pure, and in principle we would get 100% if the technical problems related to the transplantation would not be there? Or do you think we can even further improve in the future the purification of mouse hematopoietic stem cells?
If we're looking for true long-term reconstituting cells that are serially transplantable, I'm sure we can improve. But if we're even within the phenotypic compartments that we have now, even the more general sort of SLAM CD34 negative, you will find engrafting cells there, but there is a good percentage that is transient. And I believe that they're not transient due to technical reasons, but because they really cannot contribute in the long term.
Great, thanks a lot. So I have another question. This one goes to Alejo, I think. And here the question is, what is the estimated costs and financial time of such a barcoding experiment that you're describing? And what are the sequencing depth that you need for the best results?
Yeah, so we are getting better at the technique. So like, for instance, in our most recent protocol, which we hope to publish very soon, we've actually brought down the cost of library preparation, which was one of the major hurdles for this process. We brought it down to about 5 to 10 US American dollars per population analyzed, back from something that was like in the hundreds. So that's pretty good news. We're also learning how to multiplex better the library preparation protocols. So that will further bring down the costs. So in terms of preparation, you can look at about 100,000 cells or so can be analyzed with 10 US American dollars right now in our current library preparation method. Then for sequencing, at the adequate depths and roughly the costs right now of what we use is typically NeXT-seq from Illumina for sequencing these libraries, and that typically requires, because of the structure of the transposon, the fact that you have to read through the end of the transposon and into the adjacent sequence, integration sequence, that requires relatively long reads. So you can't do it with just, let's say, 25 base pair reads and things like this, both for mapping reasons and also to cross through that. And that requires fairly expensive sequencing, but it's about another $10 to $20 per population analyzed. So yeah, the setup itself is just importing the mice and having a fax sorter. So the rest of the stuff is usually handled through outsourcing these services. We typically outsource our sequencing and so on.
So in general, what do you consider actually at the moment the biggest challenges in the field of single cell or in the field of lineage tracing, the way you are doing it? And do you have any ideas on how to overcome those? Can you maybe share a little bit your thinking into the future?
Yeah, I mean, it's sort of very important to realize that this method is still requiring fax for purifying these cells and analyzing them. And that has, limits the application for a lot of cells. It also, we can't sequence, platelets that are circulating or erythrocytes that are in circulation, which is something that obviously, Joana excelled at in her work because of these fluorescent reporters. So having a way to turn those transposons into something that is perhaps expressed as RNA, and then, using that RNA, which is present in erythrocyte and is can be present in platelets, and you can sequence it from there. That would go a long way towards addressing the dynamics problem that we have. We still can't, successfully sample serially from the bone marrow this type of clones. So sampling from the blood, all lineages would be highly convenient, and sequencing costs will come down eventually, and allowing you to probably sequence at the adequate depth, millions of cells, and that would lead to your capacity to completely sort every single analyze every single barcode inside a bone at perfect resolution.
Okay, great. So Dawn, I have another question for you, and this is related to this really beautiful DSNIs, that tools that you have developed. Have you ever been able to also plug some in vivo data into the system?
Yeah, sure. I think in terms of analysis, if you have sampled the same barcodes over time, you surely can use that. So if you track cells and then analyze the barcode in blood, for example, and yeah, you can look at, and if you saw the same cell types at each of the time points, you can definitely visualize it that way. But the limitation would be if you don't have all the same cell types over time, then you have to remove that cell type for analysis, because they're not fair to plot them together.
Okay, great. Thanks for the answer, and I really enjoyed both the talks, but particularly also the question and answer parts. So I would like to extend a special thank of course, to the presenters, to the three of you, for this really great insights into particularly the technical parts, which I believe are really the most important parts actually of these experiments. I would like to thank the attendees, and I would like to ask you for completing the evaluations that you will get in a couple of days via email, because your feedback and your input are extremely important for us to further improve these webinars that we are hosting here. And with this, I would like to thank you again for attending, and wish you a great day.